Welcome back to The Explainer. I am absolutely thrilled you're joining me today because we are looking at one of the most fascinating, high stakes puzzles in all of modern biotechnology. If you're an undergrad biology or bioengineering student gearing up for your first year of graduate studies, well, listen up. You are standing right on the precipice of a massive global challenge. And today, we're going to explore a molecular solution that could honestly define your entire research career. We're talking about single cell protein. Okay, let's dive right into this because Thomas Robert Malthus laid out this terrifying mathematical trap centuries ago. Populations grow geometrically, meaning they multiply, but our food supply, it only grows arithmetically, it just plods along. Fast forward to today, and the Food and Agricultural Organization is seeing this exact trend play out with our human population and the availability of affordable, protein-rich food. We're facing this yawning gap between demand and supply, and it's leading to chronic global malnutrition. It's a modern-day Malthusian trap, and it is exactly the kind of massive, systemic problem that your generation of graduate researchers is being called upon to solve. So here is our roadmap for today. First, the global protein crisis. Second, defining single-cell protein. Third, algae, fungi, and bacteria. Fourth, the toxicity bottleneck. And finally, bioengineering the future. Section 1. The global protein crisis and overcoming the Malthusian trap. The harsh reality here is that we simply don't have the agricultural land or the resources to feed a geometrically growing population using conventional protein. Things like soy meal and fish meal are becoming incredibly expensive and wildly resource-heavy. Our traditional agricultural systems are totally maxed out. We can't just keep clearing forests and hoping for the best, right? We need a radical shift in how we generate the fundamental building blocks of human and animal nutrition. Moving on to Section 2. Defining single-cell protein and meeting the microbes. If we can't grow our protein out in some vast, sprawling field, where do we grow it? Well, the answer is inside a bioreactor. So, what are we actually talking about here? Single-cell protein, or SCP, is the protein extracted from cultivated microbial biomass. The term was actually coined back in 1968 at an MIT meeting to replace older, kind of unappetizing names like microbial protein or petroprotein. And honestly, it is an incredibly elegant solution. You take abundantly available dirt-cheap waste materials, we're talking sugarcane bagasse, sulfate liquor, or even methane, and use them as a carbon and nitrogen source to cultivate microbial cells. You harvest the biomass, extract the protein, and boom, you have a nutrient-dense food source. It's just beautiful science. Section 3. Algae, fungi, and bacteria. The comparative analysis. If you're designing a bioprocess, you absolutely have to know your organism. Now, what's really interesting here is how these different parameters completely dictate your upstream processing choices. Take algae, for example. They have a relatively slow growth rate, but their substrate requirements are beautifully simple, just carbon dioxide and sunlight. Fungi, particularly yeast, have a quite high growth rate and thrive on a huge range of substrates, especially complex lignocellulosic wastes. But bacteria? They have the absolute highest growth rate and can utilize a massive range of substrates, from agricultural wastes all the way to industrial chemical byproducts. But hey, speed isn't everything. We also care about the yield, specifically the true protein percentage by dry weight. Algae give you a solid 40 to 60%. Fungi offer a broad range from 30 up to about 70%. But bacteria? Bacteria are absolute protein powerhouses, maxing out at a staggering 83% true protein by weight. To put that into perspective, traditional crops don't even come remotely close to that kind of protein density. Plus, bacteria are exceptionally rich in methionine, an essential amino acid. Which brings us to the core academic challenge in Section 4, the toxicity and safety bottleneck, and the biological hurdles. Think about it. If bacteria and fungi grow so incredibly fast on dirt-cheap waste and are packed with all that protein, why aren't we all just eating single-cell protein right now? Why isn't it the staple of the global diet? Well, my future graduate researchers, this is exactly where the biology fights back. The first major roadblock? RNA. When microbial cells proliferate at those insane rates we just talked about, they require a massive amount of cellular machinery. That means RNA, and a lot of it. Algae are relatively safe at 4 to 6% nucleic acid content. Fungi sit around 9.7%, but those incredibly fast-growing protein-dense bacteria, they're packed with 15 to 16% nucleic acids on a dry weight basis. So why does that actually matter for us? Well, dig back into your human physiology textbooks for a second. Humans naturally lack the enzyme uricase, so when we degrade nucleic acids in our diet, the end product is uric acid. If we consume more than 2 grams of nucleic acid equivalent per day, that uric acid just accumulates in our bodies. It crystallizes in our joints and renal system, leading to excruciating conditions like gout and kidney stones. So, yeah, bacterial and fungal SCP in its raw form, it's literally toxic to humans. So to actually make this edible, our downstream processing has to aggressively reduce that RNA content. And researchers have come up with some incredibly inventive ways to do this. First, you can use a brief heat shock, around 60 to 70 degrees Celsius for 20 minutes, which actually tricks the cell into activating its own endogenous RNA as enzymes to break down its own RNA. Pretty clever, right? Second, you can use alkaline hydrolysis. And third, actually, this is my absolute favorite piece of bioindustrial genius. Researchers have successfully used pancreatic RNAs and bacterial endonucleases immobilized on crushed corn cobs to act as a chemical filter. This drops nucleic acid levels in yeast from 15% down to a totally safe 0.5%, with barely any protein loss. But RNA isn't the only danger lurking in there. We also have to purify away lethal toxins. Fungi, like certain Aspergillus and Penicillium species, can produce secondary metabolites called mycotoxins. Aflatoxins have strong epidemiological links to human liver cancer, while ochratoxin A causes severe kidney damage. Bacteria present a totally different challenge. Gram-negative bacteria contain lipopolysaccharides in their cell walls. The lipid A portion of these are endotoxins, which cause severe febrile reactions, meaning dangerously high fevers, and immunological spikes even at really low doses. These aren't just minor inconveniences. They are severe safety bottlenecks that require rigorous, uncompromising downstream purification before SCP can ever see a grocery store shelf. And that brings us to our final section, Section 5, Bioengineering the Future and Overcoming Limits. Purification and extraction are great, but they're expensive and they're time-consuming. What if we could just solve the toxicity and yield problems upstream? What if we could literally engineer the perfect microorganism? This right here is where your graduate thesis could change the world. So here's the real game changer. Instead of trying to wash away the toxins, we can use molecular biology to just turn off the factory. Let's look at Aspergillus. Researchers have mapped the biosynthetic pathways for its mycotoxins. They identified AFRR, the specific regulatory gene controlling the production of aflatoxins. By using plasmid vectors to introduce a deletion within this specific gene cluster, they successfully engineered mutant strains that are physically incapable of producing those deadly toxins. You are literally editing the danger right out of the organism's DNA. And we can also engineer for massive yield improvements. Take lysine, for example. It's a crucial, essential amino acid needed to improve the nutritive value of food, and globally we need about 180,000 tons of it every single year. By applying recombinant DNA technology to an alanine auxotroph of the bacterium Corynebacterium glutamacum, researchers pushed its lysine yield to a staggering 98 grams per liter. Through genetic recombination, we can basically force these organisms to overproduce the exact high-value proteins and amino acids that humanity desperately needs. Now, let's translate this biological theory into actual manufacturing. Traditionally, SCP is made via submerged fermentation, growing microbes in giant liquid vats. It requires continuous agitation, high capital, and produces a massive amount of liquid effluent waste. But if we shift to solid-state fermentation, or SSF, we grow microbes directly on solid insoluble polymers, like agricultural waste, with no free water. Fungi are absolutely perfect for this because their mycelial tips possess immense turgor pressure, allowing them to physically penetrate hard substrates. SSF requires low capital investment, saves space, and is practically non-polluting. It's an elegant engineering shift that drastically lowers the cost of production. And, well, that is exactly where the baton passes to you. We've defined the microbial heroes and mapped out the physiological hurdles. We know for sure that single-cell protein can offset agricultural land use and turn waste into high-density nutrition. But to make this globally viable, to make it safe, cheap, and universally accepted, we need the next wave of molecular breakthroughs. So as you head into the lab and start defining your research, I'm going to leave you with this. How will you use molecular biology to engineer the next generation of single-cell protein? The global protein crisis is literally waiting for your answer. Thanks so much for joining this explainer, and I will see you next time.